Alright, this is Unit 3, Cell Communication, and the big theme on this unit is going to be signal transduction. Now, signal transduction is a big word, again, with one of those simple meanings that we've talked about quite a bit. You heard about phosphorylation. We'll see phosphorylation again in this unit. And signal transduction is just basically how I transmit a signal from one place to another and how the cell responds to it. Now, big question we have for you is has to do with what's called differential gene expression. And differential gene expression is exactly what it sounds like. Sometimes genes are turned on and expressed, and sometimes the genes are turned off. And what that allows us to do is answer a bunch of different questions, like why do cells look different from each other? Now remember, we all started out as a single cell. Sperm fuses with egg, and then from that we differentiated into these trillions of cells that we see now. But why do the cells look different from each other? Next question is, if a cell gets a signal, why does sometimes it respond to that stimuli and other times, or other some kinds of cells don't respond to the same stimuli? And how do the cells get information about the world around them? How do they know that the temperature is increasing? Or how do they know that there are chemicals around? How do they receive those signals and then respond to them? And finally, how do they then communicate with others around them? And we've talked a little bit about what happens when a cell dies, but we haven't really focused on how they send that signal out to other cells. So the big focus in this unit is how the cells communicate with other cells. And these are some of the questions. So I think I have some zebrafish pictures here. And you can see from these guys that there's many different cell types that are in here. And we start from very simple cells that are undifferentiated until we get very complex cells that are going to be so differentiated they don't even look like they're from the same organism. So there's a couple of common features that we look at, but the big idea I want you to understand here is that this is again a way for us to understand that if we share common ancestry, then we're going to have common processes. Just like in the last unit we talked about conserved core processes, well, signal transduction turns out to be a conserved core process as well. So when one cell communicates with another one, what we get is we get a message, and that message is almost always a chemical. There's some other ways we can do it, but a message is always a chemical. And if cell B receives that stimulus, they may change what they're doing in response to it. So what we see over here is we see a chemical message being sent to cells, and it's being released by the pancreas, and it's insulin. We've talked quite a bit about that, but insulin is going to be receiving that message, and that's going to stimulate glucose to be transported into the cell. All right. On the other hand, if we release epinephrine, we may have something where we get glucose taken into the cell and then glycogen taken or stored from there. And then also here we can have glucagon, which is going to go in there. It's going to say, hey, you know that glycogen you just stored a little while ago, all those little glucose molecules? We need some more of those. So when you are low for blood sugar, pancreas is going to say, hey, we need more glucose. It's going to go to the liver. It's going to say, hey, can you go ahead and take this glycogen out of storage and release the glucose into the blood? If we got too much glucose in here, insulin's going to bind with those receptors, and then the cell is going to say, okay, I'll go ahead and take up insulin. And that can happen in muscle cells and liver cells. So those are just some examples of cell signaling. Again, these things are happening all the time, and they are happening across different kinds of species, which means that they are, in itself, signal transduction is a conserved core process. Now, we can have lots and lots of ways that they communicate. One is by simple direct contact. So when two cells are touching each other, they may be communicating very quickly. So if you see the plasmodesmata right here between the plant cells, they may even be sharing resources as a result of the gaps that are opening up in there. And then in the animal cells, you may see what are called gap junctions. So cheek cells sitting right next to each other, they may share resources or communicate with each other through direct methods. Another way we can do it is what's called cell-cell recognition. And cell-cell recognition is what you see with white blood cells when they encounter a foreign invader. They may go ahead and attack that foreign invader because the receptors do not fit the same way on them that they do on cells that are like us. So we have lots of different ways that we can communicate. One of those is called hyperlocal. Now, some other ones we have are what are called paracrine agents, and what they're going to do is they're going to send out a signal that's going to reach all the surrounding cells. So let's say that this cell has been injured or damaged. It may send a signal out that says, hey, will you please undergo mitosis because I'm dying and I need to be replaced. And so a paracrine agent is going to be a chemical that's just going to go in the local area. Now, the other kind of thing that we can have that's really local is synaptic. This is where neurotransmitters between neurons are released and they run into the synapse, and the synapse is going to allow these organs or these cells to communicate with each other. This cell may release an inhibitory or a stimulatory signal, and that may tell that neuron, the target neuron, to fire or tell it to not fire. So that's another way that we can have communication. And longer distance, now this is a little bit more difficult for people to understand, but we have endocrine glands like the pancreas that are going to release proteins like insulin, and they're going to travel through the blood, and they're going to go to target cells. Like we saw in the previous picture, those target cells would be the liver or muscle tissue. And they're going to be traveling for long distances, and so their response rates are going to be slower than these other two methods, but typically their effects are more widespread, so that lots of different cells are going to pick up that message and respond to it. So different types of messages we can have: hyperlocal, we can have local, and we can have long distance. Now, we have three main phases in the signal transduction pathway. The first one is reception, and that is typically a receptor that's going to be found on the membrane. We'll talk about the different kinds of receptors that are found on the membrane, and that is what is going to be receiving the chemical messenger, which is carrying the message. And what that is going to do is that's going to cause a pathway of molecules to kind of a cascade. So one activates the next, activates the next, activates the next, and that's called transduction. And then transduction is going to cause the cell to respond in some way. And then those responses are very characteristic. We see them all the time in cells. There's this nice little pattern of them that they can all fit into these nice little general categories with. But there's three parts to it. Receptor, where we get receptor activation between the messenger and the receptor bound to it. Then we have transduction, where a cascade of molecules inside the cell are going to be activated, and then that is going to stimulate the cell response. All right. So don't forget that pathway. This is a number one thing you want to walk out of here understanding, is this pathway. All right. Well, we have different kinds of receptors that we can look at, and you need to know each of these different kinds of receptors by appearance and then kind of by what their function is. So the first one is what's called an ion channel, and it's got a pretty simple little thing. I'm going to go ahead and blow this up just a little bit here so we can see that. In an ion channel, the signal molecule binds to the receptor and opens a gate. So an ion can pass through that could not pass through before. So you remember that ions are small and non-polar, or small and polar, and they can't pass through the membrane because that membrane is polar, just like we saw with the hydrogen protons in respiration and photosynthesis. And if that receptor is bound to that messenger is bound to the receptor, then it opens the ion channel, and we can get the ions to pass through. They're facilitated diffusion. It's just that this facilitated diffusion requires a messenger to bring them in. All right, and then the next one is called a tyrosine kinase. And what tyrosine kinase is going to do is he's going to go ahead and bring this forward here. Now, what tyrosine kinase is going to do is when the messenger binds to the receptor, causes a change in the shape of the receptor. It actually forms a dimer, two of them there. And then what that's going to do is that activated dimer is going to phosphorylate itself, and then it's going to transfer that phosphate to other molecules, which are going to cause the cell to respond. It's the beginning of the cascade, and that is a tyrosine kinase. So tyrosine kinases are going to be activating proteins as a result of these phosphorylation events. So they're going to phosphorylate themselves, and then after they phosphorylate themselves, then they're going to be activating other proteins which are inactivated until they've been phosphorylated, and then they phosphorylate something, and then they phosphorylate something, and they phosphorylate something in this little cascade until we actually get what the cell response is going to be. So that's tyrosine kinase. Now the third one, this is the most varied one of the three in terms of, I'm going to go and pull that forward for you. And these guys have the greatest variety of cell responses that are going to be associated with them. And these guys are also going to be using a different molecule than ATP, like we saw in tyrosine kinase. These guys are going to be using GTP. GTP is pretty much the same thing. It's just going to be guanine instead of an adenine that forms the base for it. But you have your G protein receptor here, and then you have your G proteins. And what's going to happen when the messenger binds to the receptor is that the receptor is going to activate the G protein. That is going to cause GTP to be oxidized, and it's going to donate its phosphate to this protein. That protein is then going to go to a enzyme. Sometimes it's on the membrane, sometimes it's somewhere else. It's going to activate that, and then it is going to cause that cascade effect, which is going to cause our cell response. And again, what's going to happen after it's been activated and it passes on its information is it's going to eject that phosphate, that GDP and that P will be put back together again, and then we can have that same process move on. So G protein is probably the greatest variety and number and type of G protein receptors that are out there, and also in terms of their function, G protein is probably the most important of the three. All right, so another thing that we're going to look at here is what is called second messengers. And second messengers are molecules which are inside the cell, which are passing on that cascade effect. And the most important one that you're going to see is what's called cyclic AMP. And you remember that ATP was a molecule that's got the adenine, the phosphate, and the adenine, and then the sugar, and then the three phosphates. That's ATP, right? So if we take away two of those ATP, we get what's called cyclic AMP. And we just kind of cycle between there. So there's the two phosphates falling off, and there's the enzyme which is going to cause that to happen, and then cyclic AMP itself becomes a key messenger in this transduction pathway. And when I say key messenger, what I mean is that it's going to cause lots and lots of molecules to be activated. So there is this huge, it's almost like a domino effect. So one cell or one molecule affects hundreds, that affect thousands, that affect tens of thousands of molecules. So cyclic AMP is going to cause this wide cascade of effects, which eventually lead again to that cell response. All right, and then the second messenger you want to know about is calcium. And calcium is a very handy second messenger for us. So here is the pathway with our G-coupled protein receptor like we talked about before, activating this little pathway right here. Then it's going to bind to a messenger. And then that messenger is going to bind to a calcium receptor, and you can see a little ion channel inside the cell where the endoplasmic reticulum is stored calcium, and then the calcium is going to go out and it's going to activate lots of different proteins, which causes our cell response. So what you see here is that calcium acts as a second messenger, and it gets basically released and taken back in, released and taken back in, released and taken back in. When it's released, it activates those proteins. When it's taken back in, they are deactivated. But in the meantime, the signal that we started with causes the cell response over here, and all the rest of this baby is just second messengers in a signal transduction pathway. All right. So second messengers are kind of an important thing that we want to know. Now, I've been talking about cell responses a lot, and those of you in anatomy are sick and tired of hearing this, but you know the 5 possible cell responses from last year that we're going to focus on. Number one is we can have a change in metabolism. So if we have thyroxine, which is a hormone that affects metabolism, that is basically going to increase the rate of metabolism of muscle cells, and that's going to increase more heat, and that's going to raise our body temperature, among other things. And that's an example of change in metabolism. We can also slow down metabolism. When you go to bed at night, your metabolism slows down as well. What the cell secretes. So if we are a messenger that might cause insulin to be released, that messenger might be glucose molecules binding to receptors on the beta cells in pancreas. And then that may affect the pancreas cells to release their insulin, and then the insulin goes and tells other cells to take up the glucose. So it affects what the cell secretes. So anytime we need something secreted from the cell, the chemical messenger will tell the cell, hey, can you secrete this for us? Cell division, they undergo mitosis. So like with the paracrine agents that we saw before, if the cell gets damaged, releases those paracrine agents, they may cause them to undergo cell division. And of course, we can also stop cell division from occurring. Right? And then membrane permeability, those are those ion channels. And membrane permeability is a huge thing when we talk about nervous transmission, which we'll get into a little bit of detail here and in greater detail second semester. And finally, what's called contractility, and that's when a nerve talks to a muscle and the muscle contracts. There's lots of other examples, but that's probably the simplest one for you to understand. Muscle contracts, that's going to cause movement, and that's going to allow you to, raise my hand like I'm doing right now, or for my food to digest, or lots of other responses. Breathing could be a result of that as well. All right, so these are the 5 possible cell responses to a chemical messenger. Don't forget them. All right. Now, what happens if we have a problem with our signal transduction pathway? In that case, you can get a disease, or you can get an illness. So let's say that you have decided to eat a tainted can of green beans, and you pick up a botulism bacteria. And what's going to happen with these guys, here's a botulism toxin right here. And what that's going to do is that it's going to be binding to proteins that are going to keep neurons from signaling muscles. And that is going to cause those muscles then to not contract. And if they don't contract, then what happens is that they are going to be, let's say it's something simple like your finger wouldn't contract, but if it's your respiratory system, then you wouldn't be able to breathe. So botulism infections are pretty severe. You want to avoid them as much as you can, but basically botulism is going to take the information that's trying to be sent from the neurotransmitters synapses into the synapse, and it's going to keep them from releasing. And as a result of that, no signal goes to the muscle, and the muscle fiber is paralyzed. Now, some people take advantage of this by shooting Botox into their forehead to give them a nice, smooth forehead. But the problem with that is you can never show surprise, and that's it could be a key human emotion. Like if you wanted to say, "Hey, oh, I'm surprised. I'm very happy to see you," they may be able to not read you very well at all because it's frozen up here. And then I just read a thing today that said that you can actually get Botox for crow's feet as well. So if your parents wanted to look better, they might put Botox right around the eye here. Some risks with it, of course, but it is a defect in the cellular mechanism that's a result of an infection. Right? And then the last one we want to talk about is called type 2 diabetes. And type 2 diabetes, next one we'll talk about, type 2 diabetes occurs when the cell no longer responds to that insulin signal. So in a normal cell, what happens if we have glucose in high concentration outside the cell, the insulin comes, it binds to a receptor, and on a muscle cell, let's say, and the muscle cell says, okay, I'll take in some glucose and I'll store it as glycogen. And so the glucose goes in. But you see here that the signal is coming. The insulin is being produced in the same concentration it was before, but now the insulin receptors are not responding to it. They're not taking in the glucose, so the glucose is staying at higher levels outside the cell. Well, that can take your body out of homeostasis. Number one, you don't get the glucose into the cell, so you don't feel as energetic. Number two, you have high amounts of glucose outside the cell, and that can have long-term consequences for cardiovascular disease and obesity and lots of other things. So type 2 diabetes is something that develops over time with repeated excess exposures to glucose. So if you maintain high concentrations of glucose outside the cell, what's going to happen to the cell is over time it's going to stop responding, and that's going to be a defect in the signal transduction pathway. Right? Now, you may have heard of allergies, and some of you may have allergies, and basically allergy is an inappropriate signal response to a chemical message, and so the body produces these molecules, or we are exposed to these molecules, and they're typically not very dangerous to us. And unfortunately for us, our body responds as if they are dangerous and releases these things that are called histamines. The histamines cause itching, they cause swelling, and they're going to cause general just pain. People who have allergies know what I'm talking about, right? And antihistamines do this super cool thing. What they're going to do is they're going to bind to the receptors. So whatever is causing the histamine response, or basically, sorry, they're going to compete with the histamines to block the receptors. So they're going to be competitive inhibitors for the receptors. Now, we talked about those before with enzymes inside the cell in the last unit. Now we're talk about them the same way with receptors on the membranes of cells. So when you take the antihistamine, the histamine can't bind to it. You don't have the allergic response because your cell's response is muted or blocked by these competitive inhibitors. So as long as you take the antihistamines, then you don't have the problems. Once you stop taking the antihistamines, if the allergens are still being produced, then you're going to have problems. All right, so that is it for this unit. And remember, our key idea here about signal transduction is that cells are going to respond to things in specific ways, the signal transduction pathway, and that cells can communicate over both short and long distances and hyper short distances, and this is going to allow us to have a response to all kinds of different stimuli. We can communicate with the cells nearby us, they can communicate with us, and we can help maintain homeostasis for the entire system. Thank you.